Perfect. Yeah, great. Thank you so much. Thank you, uh, Elizabeth, for introducing. And I know this is afternoon, and of course, this is Friday. So I am not going to bore you too much, and we will be to the point actually. And of course, you are always free to interrupt me actually if you have any question in the middle of this presentation. So welcome everyone and thank you. And I'm going to talk about biology of the flash radiation and its comparison with conventional radiation. And the concept of flash radiation is quite new. It is still not in the clinics, but of course, I will discuss actually some of the clinical trials which are. So let me put into a presentation mode. Uh, okay. So this is how we are going to cover this whole presentation. Actually, that therapeutic index of the conventional radiation versus the flash radiation, and what is the historical background of the flash radiation and how it started actually this whole concept and when it gained the steam and where we are now. So we'll give little historical background and then radiotherapy and flash effect. Actually, what is this and how it is different uh, from the conventional radiation, which is in the clinic. So that will give us a comparative analysis actually of these two mode of modalities actually of radiation. Then flash hypothesis and molecular mechanisms. So again, this is pretty new actually advancement in the field. There are some existing hypotheses, but those hypotheses still there are a lot of questions surrounding those hypotheses. And what is the new hypothesis and what we are doing actually to propose actually if there is something new. So always actually this is quite exciting actually that what are the existing hypotheses and what could be the next which can be uh, the breakthrough in the field. Then we talk about the flash radiation and mitochondrial metabolism because mitochondria is the source where ROS production takes place. So we will discuss about this. And then there are some few preclinical studies actually which has been completed. And those preclinical findings are quite exciting actually if we will discuss about this one. And then also the tumor killing effect of the flash radiation and then some of the clinical trials which have recently been completed. We'll discuss about those clinical trials and then, of course, the conclusion. So, so the background and then the flash actually was was quite um, existed actually long back, actually in 1959, when they proposed actually this whole concept of the flash radiation. So, but the advancement over the time in the field, uh, delivering the beam and imaging, um, it quite uh, basically happened in such a way which is now very immensely important to conformal radiation therapy, and where the radiation uh, uh, therapeutic index can always be enhanced through the precise targeting of the tumor while maximizing the collateral, minimizing the collateral damage to the normal surrounding tissues, which is the center point actually of this presentation. Um, so the precision radio therapy has allowed us to develop radiosensitizers, radioprotectors, which are designed to differentiate the killing of the cancer cells uh, versus normal cells. So the biological effect of radiation, those to organ at risk surrounding the tumor uh, are the major dose limiting constraint in existing radiotherapy, actually, the way uh, patients are being treated. So let me, okay. Uh, okay, so now this can, this means that the tumor cannot be completely destroyed when we are treating these patients with the radiation treatment under current circumstances. Because what is happening that when you are calculating the dose uh, to destroy the tumor completely, you are also affecting the normal tissues, and then normal tissue damage is always there. So in order to, and then Dr. Carrier and some other speakers, they also shared that what is the percent chances that the normal tissue will get damaged when you are targeting the tumor. So there's a very fine line that how we can save. You can completely destroy the tumor tissue or the tumor and then save the normal tissues actually from the radiation induced damage. So thus, this reduced damage to healthy tissues has always been the topic of interest of radio therapy research. And modern radiotherapy technologies such as helical tomography, and then intensity modulated IMRT actually, and then for that matter also proton radiotherapy can reduce the damage to healthy tissues to some extent. And the example is here that when we are treating these patients in the brag peak, so what is happening that you are concentrating on the tumor, but what is happening that of course you can minimize this, but you cannot avoid the normal tissue damage. So the advancement in the field, proton therapy is okay, but still you can minimize the damage, but we cannot avoid the normal tissue toxicity. So then it comes, so what is the balancing act actually when we are treating these patients? So the balancing act is that when you are trying to get the full tumor control and then destroy the completely the tumor, then also you are going to damage the normal tissues. So that fine line of basically that therapeutic window is quite constrained. Actually, it is quite tight actually when you are talking about the normal radio therapy. So here, the tumor control is there, but normal tissue damage is also there. So that fine line is very, very important actually in treating when we are treating these patients in the clinic. So now back to FLASH radiation therapy. Then it was 1959, and then some of the scientists they started doing some experimental work actually in bacterial suspension, and they trying to see that suspension. They found that okay, when it was oxygen-rich conditions, then they have a sensitivity to radiation, and then of course this premise began to research on ultra high dose rate radiation actually at that time. Then later on, some of the experiments were also performed in the HeLa cells and in 1963 and found that the dose rate reached to the high point, the radiation sensitivity decreased with an increase in the dose rate. Um, Now, after this, they also performed some of these other experiments in presence and absence of the oxygen. And here in this one, they performed the experiment, second set of the experiment using nitrogen. And then when they were using the nitrogen, they failed to show the same decreasing sensitivity to the radiation. And that basically suggested that radiation resistance was related to the oxygen effect. And that gave this idea for the oxygen consumption and other sort of hypothesis surrounding this oxygen. Then later on, Barry and some other scientists, actually in 1969, they also performed some of these shortening pulse delivery time and proposed that oxygen depletion reduces the production of radiation-induced free oxygen radicals, and thus avoiding the normal tissue damage. And it was like in 1969, which was way back. And then thus these free radicals generated by the differences in redox metabolism between different tissues is a valid indicator for defining the flash effect here, because we know what is the difference in the cancer cell metabolism and then normal cell metabolism. That redox metabolism gave this idea about this flash effect. So what is the flash radiotherapy and what is the flash effect? So the flash radiation is delivered at ultra high dose, actually 4 gray per second, to a total of, in the total matter of 100 to 200 milliseconds. And that basically time um, is very important actually when they deliver these doses, because what is happening, that will allow to treat the tumor without inducing drastic toxicity to the surrounding normal tissues. So they are minimizing the time and increasing the dose when we compare with the normal conventional radiation. So the flash effect is the biological phenomena, and that consists in a remarkable sparing of normal tissues while killing the effect on the tumor, which is the main aim actually of this flash effect. So flash radiotherapy is a novel technique in which the ultra high dose actually rate is delivered at 40 gray or higher per second, which is delivered to the tumor tissues and which cannot damage to the normal tissues. So that is where the ultra high dose delivery within short span of time in milliseconds is the key actually that flash effect uh, 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 is taking place. So some of these in vivo recent studies on this ultra high dose rate actually has the potential to spare the normal tissue without sacrificing tumor control. And that will present some of these evidence actually when we are discussing this one. So this one is a difference here. We can discuss a little bit about these plots actually where possibility of widening the therapeutic window is higher in flash radiation. So this is a conventional radiation when we discuss about the therapeutic window, where we saw the tumor control and normal tissue damage. And there's a very short window actually here where we are very fine line we are always taking care of, but still there is a damage 5 to 7% to the normal tissues actually when you are delivering the dose which we are thinking can kill the tumor at 100% level. But when you are discussing the flash radiation, then always we are seeing here that this particular window actually of normal tissue damage and then tumor control can be widened actually. Since what is happening that here, there is a very minimum or no normal tissue damage, you can always deliver high dose rate more than 100 gray per second actually, or millisecond, and that higher dose can completely kill the tumor and that it can enhance the therapeutic window. So that is basically possibility of the therapeutic window enhancement, and this is where the flash radiation can change this whole game actually of radiation therapy actually in the clinic. Now, so in order to enhance this therapeutic window, what are the dose rate which flash can deliver actually? So you can see that the high rate of the flash radiation is much higher. Let me manage this. Okay. So
Are
the
there
flash
any
radiation
issues?
is 400-fold more rapid than the conventional radiation when we are treating these animals in the preclinical studies, actually, or in vitro studies. The flash RT dose is delivered in several nano to milliseconds. That shorter span, much shorter than conventional radiation. And recent experiment has revealed that the hypoxic normal tissues cannot get reoxygenation in that short span of milliseconds, which controls the actually process performed in such a short span of time that normal tissue get protected against the radiation damage. So that is one of the thing that, okay, there's a very short span of time, milliseconds, where that millisecond actually where the hypoxic conditions are uh, there, and then it can create hypoxic condition and where it can protect the normal tissues, but at the same time can kill the tumor tissues actually. So that we'll discuss how it is happening. So the tumor cells have altered metabolism. We have seen it. They altered glycolysis if you compare with the normal cells. And then that aerobic glycolysis is producing huge amount of ATP for the tumor growth and fueling the tumor. And at the same time, there's also huge amount of the ROS production actually in the cancer cells. And that is basically, that's why the cancer cells are susceptible for the ionizing radiation induced cell death. So the tumor is also characterized by well-established immunosuppressive tumor microenvironment, and that some of these solid tumors, like pancreatic cancer, they are hypoxic and then highly fibrotic. So the immunosuppressive tumor microenvironment also leads to the radiation resistance, and when it is exposed to conventional radiation, it induces high rate of tissue fibrosis and senescence. So tissue fibrosis and senescence is caused because of the conventional radiation dose, which is delivered at a very slow rate. So, and that basically also is one of the issue we'll discuss about this fibrosis and then cellular senescence, actually, when you're talking about conventional radiation versus flash radiation. And then the other thing, we have seen it, other people, they have also seen it. The TGF beta pathway is upregulated when the conventional radiation is delivered to the tumor tissues. And that TGF beta upregulation, um, compared to the flash radiation, is very, very less. And we will see that one. So the level of TGF beta and then that pathway, which can lead to the SMAD4 and then radiation resistance, is almost very, very low if you compare with the conventional radiation. So these are some of these flash radiation. Actually, these are some of the preclinical data are coming in now about the mechanism of the flash radiation, which we will further discuss, and the role of the TGF beta in designing some of these therapeutic target in treating some of these solid tumors. Um, so So what are they, how it is delivered actually, the flash radiation? So we can see that when we are delivering here the 10 gray of conventional dose radiation at the rate of zero point 0.1 gray per second, then it is taking almost 100 seconds. But when you are delivering 50 gray per seconds, it is basically 1.8 into 10 to minus 6 seconds, which is way, way less actually if you compare with the conventional radiation. So you are minimizing that window of delivery of that flash radiation dose and maximizing the dose delivery. And that is where that is very, very important actually when we are talking about the conventional dose delivery versus flash dose delivery. So this is where it also affects the outcome of the treatment. Now, if we are talking about the flash radiation and its mechanism, huge amount of animal experiments have been performed actually so far, and they have been compared with the conventional radiation actually induced damage to healthy tissues, and and also the first clinical treatment actually happened in the cutaneous lymphoma, T-cell cutaneous lymphoma in Switzerland, and they used actually electron-based flash treatment, and then they saw the anti-tumor effect um, uh, when they compared with the conventional radiation. However, there was not very much difference in conventional radiation versus flash radiation. However, when they performed actually using proton radiation, then of course the new studies actually is showing very significant difference in the treatment outcome actually of those uh, uh, flash radiation versus conventional radiation. So um, so under this first, actually, that test actually happened quite long back, and after this, couple of clinical trials have been completed, which we will discuss in the end actually of this presentation. So the hypoxic normal tissues in such a short period of time cannot be detected by the response of the hypoxia-mediated marker, but there are evidence that there is a transient hypoxia when the flash radiation is delivered in the normal tissues, and that basically that transient hypoxia is protecting the normal tissues and damaging the tumor tissues. So some of these things has been done actually, these preclinical models, which are mice, zebrafish, and then also mini pig, and then cats, and so on. And they have shown all the reproducible effect of the flash radiation. So how the flash radiation has shown the improved therapeutic ratio with improved normal tissue sparing while maintaining the same tumor killing effect as compared to the conventional dose rate. So the good thing is that it always maintains the same level of tumor killing if you compare with the conventional radiation. So absolutely, if we cannot compare, but we can always say that the normal tissue toxicity is almost not there when we are treating with the flash radiation, and that is a quite a significant statement. But maintaining the same tumor control but sparing the normal tissues toxicity is a quite game changer actually if the flash radiation is basically approved to use in the clinics. So further, so these flash radiation actually to understand further about these dose actually and then clearly challenging the flash radiation where chronic low dose exposure were routinely delivered in conventional radiation documented to ameliorate the adverse effect of the radiation. So if we compare for a second the fractionated dose delivery of conventional radiation with flash radiation, the effect, the purpose of fractionated radiation was the same, that we are fractionating radiation to minimize the dose. So that is not causing normal tissue toxicity. And then you are extending that window actually of the treatment. But in case when we compare with the flash radiation, then of course you are delivering that dose of 40 gray in a matter of milliseconds, and that is effect basically is the same. That you are minimizing the time, delivering the high dose, and achieving that goal of not causing too much radio normal tissue damage, but killing the tumor. So looking at this, basically, we have seen, and other people have also seen that the clonogenic survival cannot be used to study the flash effects and its comparison with the conventional radiation. And we did actually, and we did not find any difference in the clonogenic survival of these um, uh, cancer cells when we treated both with the flash and then conventional. So that statement actually is right. So what is the, if we talk about now the molecular mechanism of flash radiation? So since 2014, actually, when there was a these French scientists actually they started these experiments, preclinical experiment, then this whole uh, fresh radiation field actually came into again into the full swing from 2014 onwards, where they started understanding the role of the flash radiation and how the flash can be used actually to treat actually the tumor, sparing the normal tissues. So the hypothesis actually and the mechanism of this existing mechanism of flash radiation are currently there are two acceptable hypotheses, but still those hypotheses are in questions, and therefore still the field is in the beginning phase. Actually, it is not has been taken off where we can say that okay, these are the mechanism of the flash radiation. This is how it is damaging the DNA. This is how it is not damaging normal tissue toxicity, and then no side effects, so that we can convince the clinical world actually, so that we can bring it to the clinic. So the flash radiation can potentially change the paradigm of radiation therapy. The biological mechanism behind the flash effect is largely unknown. And then there are so many these differences in the early physiochemical steps between flash radiation and conventional radiation had not yet been formally investigated. And there are still many unanswered questions surrounding, of course, the mechanism, and then the those existing hypotheses actually, which are still under question mark. So, what are those hypotheses, current hypotheses, and what is the mechanism of the flash radiation? So, the current hypothesis is the high rate of free radical recombination and diffusion, and then number two is oxygen depletion and hypoxia. So, number one, actually, the free radical recombination and diffusion was speculated to elicit um, higher instantaneous free radical densities that could facilitate their recombination and alter diffusion of free radicals and the production of radiolytic species and the downstream damage. And this hypothesis was quite strong actually, because in 2019, actually, Monte Gruel et al. In 2019, they performed one of the experiment, and that experiment was compelling experiment evidence in favor of the diffusion recombination hypothesis, where they showed here actually that they measured the hydrogen peroxide. So they treated the water with flash radiation and conventional radiation, the same dose. And then you can see that these are the dose versus hydrogen peroxide actually product release. Then they found that when they had this, then at the 4% O2 water level, the conventional radiation has had much more generation of ROS actually than the flash radiation. So that hypothesis, and that was quite convincing, that when you're treating this water, the ROS production with the conventional radiation was significantly higher than the flash radiation. So then later on, the flash radiation could have the ability to elicit transient volume-dependent drop in oxygen tension, and possibly sufficient to cause radioprotective hypoxic state. And this was also basically based on the oxygen enhancement, classical oxygen enhancement ratio, which is between 2.5 to 3 actually. So this was where that transient hypoxia uh, that uh, also came into play actually, about explaining um, these things. Now, so this one is again in the brain tumor, where they found actually they, when they treated with the dose of the conventional radiation and then flash radiation, they found the level of ROS actually treated radiation was significantly low than the they treated with the conventional radiation, and that basically low level of the depletion of the oxygen and transient local hypoxia in the brain could protect actually from the normal tissue toxicity and then radiation-induced damage. So that was quite a significant finding when they stated that there was a low ROS actually production when they are treating with the flash radiation. So then the other one is hypoxia hypothesis and oxygen depletion hypothesis. And this hypoxic is based on the ROS. That okay, the hypoxia hypothesis was generated in the performance in vivo experiment, where they found that under hypoxia was eliminated when they again treated this carbon breathing actually, which could oxygenate the whole brain actually. When they were treating with the with this flash radiation. So they could reverse the effect of this flash radiation actually by oxygenating the level of the brain. So that again was quite convincing that okay, this reverse actually carbon breathing can definitely reverse the flash effect. It was quite significant finding actually here actually in 2019. And then some of the recent experiment again, like oxygen, measuring the oxygen tension in vivo, and how provided convincing evidence that radiolytic hypoxia induced by flash was sufficient to provide any biological benefit. So these recent measurements also fit past the data finding that doses approaching 100 gray are typically required to elicit 3% drop in oxygen level, um, that those that are far above those necessary to observe the in vivo flash effect. Um, so this is also quite significant actually finding in the recent some of these research. Now, the other hypotheses also are still there, like which they have put forward, protection by stem cell niche actually in the, so 2019, the scientists, they said and they proposed that a stem cell niche, flash radiation also protect the stem cell niche actually, and which may involve tissue specific hypoxia. So the presence of the stem cells in the brain electron flash RT was reported to preserve the neurogenic niche where compared to the conventional radiation. So that hypothesis is basically a little bit controversial. Because why it is controversial? Because if we are thinking about the role of cancer stem cells, where we are saying that cancer stem cells are responsible for radiation and chemotherapy resistance and treatment failure in cancer, and if the flash RT is protecting them, then of course this is controversial hypothesis. So it is basically not acceptable actually, uh, this hypothesis currently actually. So then one potential factor that could contribute to the modified immune response is also convincing actually, which is eventually lead to the flash effect, could be reduced circulating immune cells killing at flash dose rates. So some of these scientists, they use the conventional dose rate and then also the flash dose rate. And then when 2021 actually, they treated under both this, and they found that under conventional radiation, they found that there was 90% death of these immune cells when they compared only 10% immune cell death actually treated with the flash radiation. So that basically looks little convincing that sure the flash radiation could be not affecting too much immune cells death, and therefore it can also mount robust immune response. So that is also, some of these experiments are underway to prove this actually, that hypothesis. So the flash radiation has a strong protective effect on the immune system. That has been proven actually in many preclinical model, and this result might be beneficial in reducing the radiation-induced toxicity in normal tissues. So that could hold the water actually, that particular hypothesis. And based on the immunotherapy, actually, there could be a combination modality with flash radiation with immunotherapy, some of these checkpoint inhibitors, where it could be successful in treating some of these also tumor, which can activate the immune response. So that looks quite, this current the hypothesis looks quite convincing. So now, the flash radiation can reduce the complication caused by conventional radiation, and that is true. Protect normal tissues from the apoptotic induction, and that is also because there's no normal tissue damage. So versus conventional, so definitely there's no apoptotic uh, caspase induction in the normal tissues. In 2019, Giordani also et al. Actually, they investigated mechanism of proton flash RT in mice model, and they reported cytokines inducing apoptosis of T lymphocytes were increased after treating with the conventional dose, but decreased actually after treating with the flash radiation. So this is another reproducible data actually on the immune cell and then the flash radiation. The further, actually, they also used actually some of these markers like CD34, which is the epidermal stem cell marker. And then in that epidermal stem cell marker, they found that when they're treating with the flash radiation, the CD34 was still basically expressed this one. So and it has minimal effect on stem cell consistently with what was already uh, reported for the neural and intestinal stem cell. The other possible hypothesis, which are also proposed, the flesh depletes the critical compound impacting the radiation response or does not generate the radiolytic production of paracrine mediators. So consistently, reduction of the inflammatory and fibrotic factors has been described in normal tissues exposed to the flash radiation. And Favardon, actually, he's one of the pioneer in recent flash research, actually. And we will discuss some of his preclinical data in this one. So the flash effect, actually, when we are comparing with conventional radiation, is definitely has a very, very significant effect in improving normal tissue toxicity, but at the same time, it can also kill the tumor. So that is basically very important. The other important issue is that how does flash RT and mitochondrial metabolism um, um, is affecting actually. So the flash radiation, it has been proposed also this hypothesis that flash radiation is affecting mitochondrial lipids and then also affecting the reverse electron flow of these electrons into the mitochondria. And that again is under basically reproducing actually the data if only one group actually has reported this particular finding, that reverse electron flow. And the reverse electron flow is, if you compare so this is mitochondria and mitochondrial membrane where oxidative phosphorylation takes place, and then ATPs are generated. So when we are treating with the conventional radiation treatment, then we see that these electrons are channelized, and then oxidation reduction reactions happens, and then it goes into the terminal acceptor, and then of course the ATP molecules are produced and so on. But in this case, what is happening in flash radiation, they have found that in stage 3 cytochrome C and 4, these are the electron transport components where the electrons are not flowing, and they are basically accumulating at this SDH actually at this level where it generates the fumarate. So that reverse electron flow and reverse electron accumulation is also reported actually recently. And then so reverse electron flow favored under hypoxic conditions drives the accumulation of ubiquinol and succinate dehydrogenase. So this one again has not proven yet, but the scientists, they're trying to reproduce this data actually on reverse electron flow in mitochondria when they're treating with the flash RT. Now, uh, what I was talking about, some of the preclinical studies actually we can discuss involving these flash radiation. So this one was published actually in 2014, where the French group actually Favardon and then Voisin, they reported actually that okay, ultra high dose radiation increases um, uh, the differential response between normal and tumor tissues in mice. So they found that when they treated actually the tumor animals with different doses of the radiation, this is control. Then 17 gray conventional radiation, X-ray, and then of course 17 gray flash radiation at 4.5 MeV, and then this one is 30 gray flash radiation. And in the end, what they tried to compare is that they tried to see here that in the conventional radiation treated these tissues, they had huge amount of the fibrosis versus when they compared with the 17 gray flash radiation actually here. So they still look fibrotic growth, you can see it here, but in the flash radiation, they did not observe any fibrosis in these tissues. So that gave us the further the assumption that flash radiation definitely is not causing fibrosis because the level of TGF beta is suppressed actually in those samples. And then when they treated with radiation, actually those with 40 gray per second flash radiation, and that those short pulses were 500 milliseconds. And when they compared with conventional, they found quite a significant fibrotic tissues. Now, so this is also they compare the level of the TGF beta actually. So you can see that this one is the control, and then this one is the conventional uh, uh, treated actually tissues, 17 gray, and this was 17 gray plus treated, where they found the low level of TGF beta. So that was quite convincing, saying that, okay, low level of TGF beta is always suppressing the fibrosis. And then this is where this finding actually published in 2014, quite significant, and it is really breakthrough in the field in the clinical research. Then further, there was another preclinical finding that was the orthotopic model of the glioblastoma. And in that one, they also compared this uh, the tumor growth actually with the conventional treated versus 14 gray flesh treated. So when they compared this, they found that it is quite the tumor control is quite comparable actually. So they wanted to show that okay, absolutely there was no compromise in the tumor control, but absolutely there was also no normal tissue toxicity when they are treating with the flash radiation, and that was again a quite reproducible result and data when they presented actually in clinical cancer research recently in 2021. The another preclinical study was on the mini pig when they uh, were trying to see the normal tissue toxicity actually in these animals, and they treated with the 34 gray of and then 31 and 28 gray of this conventional radiation versus flash radiation. So now you can see that in terms of the appearance of these tissues, actually, uh, they found quite a big damage actually when they saw and then when they treated with the conventional radiation. At the same time, when they saw it, when they stained it in after 36 weeks of exposure, they found that, of course, the flesh treated, those tissues were quite intact when they compared with the conventional radiation treated these tissues. So a lot of these tissue remodeling happened actually in the conventional uh, radiation treated some of these tumor tissues. So that was again quite a significant study actually at the preclinical level actually of the mini pig. So that again, this data, preclinical data is quite significant in terms of the normal tissue toxicity. Then they did another quite elegant preclinical study on squamous cell carcinoma of nasal plenum in the cats. And when they're treating these cats with the high dose of the conventional radiation, then they have huge amount of side effects. Then the quality of life is always compromised actually in those patients or those animals, those were treated with the conventional radiation. But but you can see actually when they treated with the between 25 to 41 gray flash radiation, these cats actually they showed no normal tissue damage. They were still okay after the treatment, after 14 months actually of the treatment. And then, of course, when looking at those animals, the quality and everything was quite comparable when you compare with the huge amount of the radiation induced tissue toxicity. So that's one again quite important actually preclinical study in these cats. So the benefits of single dose plus radiation. So the benefit of single dose plus radiation, as we discussed, that we are treating these animals in the single dose of, let's say, 40 to 100 gray per second, then that single dose can destroy the tumor, but always protect the normal tissues actually at that high dose. So you have a very high therapeutic window when you're comparing with the conventional radiation dose. Actually, that window is basically giving us quite a uh, leverage actually to adjust the dose in flash radiation and completely kill the tumor. So this is where, in terms of the killing the tumor and recurrence, that would be quite important actually finding where we would like to see that if we are treating with the radiation and then how often the recurrence will happen, that is also quite important because what is happening that in normal tissues, we have seen the cancer stem cells are still sitting there. If we are treating with radiation, they are not going away. So maybe flash radiation in combination with some other modalities can also kill these cancer stem cells where the recurrence could not be the problem actually. So this is again, it is totally the field which is totally right now under investigation actually in some of these things. So now the tumors are definitely a huge amount of the assurance came when they saw that the tumor killing effect is the same. So there's no uh, that flash radiation is not uh, affecting the tumor tissues. It is damaging the DNA, and it has been found that there's a huge amount of complex DNA damage when they compared with the conventional radiation. So that complex DNA damage, again, is also very important that cancer cell cannot basically repair that damaged DNA, complex DNA damage in the cancer cells. Um, Now, what are the other mechanisms which are still um, scientists are trying to understand? Um, they are trying to see the complex DNA damage, and if there's a complex DNA double strand break or multiple those complex DNA damage, then whether the cell is able to repair that damage or not, it is still quite open actually that it could be involved actually there. The other one is, of course, the immune hypothesis that when some of these animals were treated with the flash radiation, they found that the immune cell damage was minimum when they compared with the conventional those treated samples. So that is again very important. The third one is protein targets. There are we were talking about the reverse electron flow in mitochondrial membrane, where succinate dehydrogenase is involved. So that basically also maybe structural, some of the proteins might be the target of this flash radiation, which can also block actually in the tumor cell and then can instantly kill the tumor cells. So then lipid peroxidation actually that we did not cover, but it has been found that the lipid peroxidation in flash radiate treated samples, they found much higher of the lipid peroxidation than the conventional radiation. So that is another open field actually where the scientists are trying to understand that what is happening when these uh, animals are treated with high dose of flash radiation. This is also quite interesting actually, this one. And then of course, the other one is cancer stem cell niche, where we presented some data where the cancer stem cells were not affected actually, and then they said that it protects the stem cells. And then the de-differentiation of the stem cells then comes to the cancer stem cells. So if the flash radiation cannot destroy these cancer stem cells, then of course it is always controversial when we are saying that there's a huge role of cancer stem cells in cancer. So that's again open actually question here. And then of course the transient hypoxia oxygen depletion, and that is the best accepted hypothesis right now which we have to explain the flash effect. But there are still some questions actually around this mechanism. So that's the one. And then some of these clinical trials actually which has been completed recently, and and then one of the clinical trial which is recently completed actually here in Cincinnati. So this one is, of course, the was done in University of Lausanne, actually, Switzerland, and which was a multi-resistant cutaneous lymphoma, and that was they gave actually 15 gray, 90 milliseconds electron the flash actually, and then of course they compare. They did not find any huge amount of um, positive data, but they found it is quite comparable. And of course, the long-lasting effect actually of the flash effect was there actually in that patient when they treated with the electron flash. The other one also recently happened. This one is a non-randomized trial actually in Cincinnati. And then where what they found that they treated actually the bone metastasis, and then they 10% they treated with the flash radiation, and then when they treated with the flash radiation, they found that those actually 40 gray per second and they completely found that absolutely there was no normal tissue toxicity, and uh, there was absolutely no inflammation at all. So they have been following actually quite a few weeks to months after the treatment, and that basically a quite very encouraging clinical trial which recently finished actually in Cincinnati. So with that clinical trial, there's a huge hope that, of course, this we are close enough actually to bring flash radiation to the clinic. So, in conclusion, actually, so what we have learned actually from the flash radiation, all the preclinical research, and very few clinical trials. So that basically radio-resistant tumor increasing total dose without the associated surrounding tissue toxicity. So we can treat actually radio-resistant tumor by dose escalation actually. So there's no window that, okay, we are looking for the therapeutic window. We can always enhance that therapeutic window and then treat actually some of these radio-resistant tumors. Treat diffuse non-localized tumor where conventional radiation cannot deliver tumoricidal doses. And then, of course, the treat tumor where radiotherapy already offers good local control, but without the side effects typical of conventional radiation and re-irradiation actually that we have seen in many malignancy. Actually, we re-irradiate actually these tumors, and that re-irradiation is again brings actually that tumor to the radio resistance level, and then that is basically can be taken care of if the flash radiation is in the clinic. So with that one, thank you very much. And if we can discuss some of these questions, that will be great.
Well, thank you, Dr. Shukla. You're ahead of time, 5 minutes, so we have 5 minutes for questions, if there are any. I don't see anything posted on in the chat, so perhaps, so maybe they will be coming now. I don't know. I don't see anything here.
Sure. So, I mean, everyone is always welcome. I just wanted to say that we are working on flash radiation. We are also working with the using animal models. We are also working using organoid models. So we will see actually if there are new mechanism we see, or if we see a robust immune response actually using flash radiation. So that basically which we are doing. So everyone is welcome in the lab, and we can further discuss about some of these aspects of the flash research actually in our lab.
Well, thank you. So we have 5 minutes, 4 minutes. Yeah, we have 4 minutes. So perhaps if there are no questions, yes, if we don't have any questions, perhaps we can have Dr. Zeman come bring her presentation in. Thank you, Dr. Shukla.
Thank you, Dr. Elizabeth.